And so this was the question at that point. And then what would be the follow-up question? Well, if we now did a dose-finding effect that was an open-label study, right? Then the question would be, what happens when you do a double-blind placebo-controlled study? And we got an NIH grant to do that, which is what I summarize here. And I think that this illustrates a bunch of things. One of the things that illustrates, if you look at the number up on the top, is that the number of patients that we screened to do this study, which is a randomized double-blind controlled study to see the effect of carnitine supplementation on fatigue in patients that have 6 months of life expectancy, we looked at 1,148 patients. We ended up with 60. 60 were eligible. And from these 60, then we accrued 33. We randomized then, I can see here, but about 17 and 12 in the two arms. And then these patients went through the supplementation. And at the end of the day, we ended up with, in the placebo arm, with 12 patients and 17 patients. So this tells you a couple of things. First of all, this is a proof of principle study, right? If it works, you can say that it works for a very selected population, because we started with 1,000 patients that we looked at, and we ended up only with 60. So this is one of the things. The other thing that it tells you is that this type of research is very, very difficult. So how come, if you look at 1,000 patients, you can only identify 60? What's going on? Well, these patients are dying. So these patients don't want to be bothered. The patients, when you approach them and you start presenting the study to them, they ask you, oh, so is there a chance that I'm going to get cured? I say, well, not really. This is only to get you a little bit better. But I'm almost not moving here, so why do I want to be able to go to the bathroom? So they don't want to be bothered. And the other thing that is very interesting, but this is from the perspective of the data analysis and for picking up a difference and an effect, is that usually what you do, if you look at the studies that we normally talk about in pain, what you do is when the patient is accrued and before starting the intervention, you have 1 or 2 weeks where you collect data on pain, right? And you want to see that the pain is stable. If the pain is not stable, you adjust the medications and the pain gets stable, you collect data, and the patient has to be 1 or 2 weeks where the pain is going to be, 5, 6, whatever you define for that particular study. In patients that are dying, like these patients that we have here, that I would say that 90% of these patients were on hospice. So by definition, because of the reality of the system now, these patients have a survival rate of 34 days after being referred to hospice, right? So the patient is referred to hospice. Usually they come at the time that they are referred, they are in the hospital, or they come in the hospital because there is some imbalance or the pain is not well controlled. Patient comes here. We talk to the patient, we do the accrual, so maybe 3 days go by. So this patient has now 30 days of life expectancy at the time that they are accrued for the study. These patients are deteriorating, right? So the fatigue is going to be increasing, right? It goes up. So, and you are going to try to detect a signal there on something that is moving up. So the effect that you have to see has to be even steeper than that. So it's just very difficult. How can you pick up that signal? So probably the solution to that is, one, doing with patients that are more stable. So rather than patients that have a life expectancy of 34 days, do it with patients that have a life expectancy of a year or 6 months at least. So there is, at least you can have the patients at the beginning where they are more stable, and then you can collect data that are a little bit more meaningful at that point. The reason why we did it this way is because the NIH felt a little uncomfortable about doing this study on patients that had a longer life expectancy, because we didn't know exactly what the effect of carnitine was going to be. Even though there were data that it was suggested that it was safe, there was a possibility that you are feeding into the, let's say, the cancer cells, right? And the cancer was going to advance in a more rapid manner. So there was a little bit of fear about that. And so this is the end of the study, is the data. But basically what we found then is that, as I said before, that there was an improvement in fatigue, there was an improvement in sleep, also an improving mood, as we have seen in the open label study. So this was very interesting. the reason and the point that I want to make that is, again, very educational and helps you to understand the way that we look at the data, is that the way that we reported these studies, it was negative. And the reason why it was negative is because when you do an intention to treat, meaning every patient that you accrue in the study is going to go into the data analysis, regardless of how far they went into the study. Many of these patients died in the middle of the study. So if you do that, and you got patients that are dying left and right, and you are looking at improvement in fatigue, it's not going to help. It's not going to work that well. So when we did this, we didn't see the improvement in fatigue. Now, then when we did a per protocol study, meaning what we meant was to substitute the carnitine for 2 weeks in these patients. So we used only the completers, patients that completed the study. When we look at these patients, then we saw an improvement in fatigue and other outcomes. So that's very important to know. But what it was good then is that it helped us to design the following study. So this was a small study, proof of principle study. There is a suggestion that maybe carnitine can be helpful to these patients. So what we did afterwards was to design a larger study. We got ECOG involved, and these are the centers that participated in the study, and the N reflects the patients at the different institutions accrued, and the total of patients was 376 that we were able to accrue. So we went from 12 plus 17 to 376. So it's a huge change in the study. And what we did is the same idea, but a little bit different. In the other study, all the patients were carnitine deficient. That was one of the inclusion criteria. In this study, we took every patient with fatigue and cancer, regardless of the carnitine status. So the patients were accrued, and then we measured the carnitine later on. And we did a post-hoc analysis trying to see if the carnitine level had anything to do with it. So we ended up with 199 patients in one arm and 187 in the other arm. And as you can see, there was an improvement in fatigue. Look how nice. This is the carnitine supplementation. 4 weeks, improvement, statistical significant. Week 8, improvement, statistically significant. However, when you look at the placebo arm, So placebo can kill you. It's terrible. How can you make any conclusions with this placebo? So when you compare these two, then the effect that you saw, it goes away. And this brings us to the last point of this presentation, which is what can you do with the placebo response? One is ignore it, just do open label studies. It doesn't help you that much. But what you can do is to decrease or control the placebo effect to some point, to some extent. So what some people are saying is the one way that you can work on that is to do longer trials, because if you have the expectation that you are going to feel better when you do something, when you take a pain medication or something for fatigue, and you are very excited, the first week, the second week, very excited, by the 4th month you are not excited anymore. And then, it works, it doesn't work, I don't care. And the problem with that, even though the placebo effect goes down, is that you have a higher dropout, because people just don't have the energy, don't have the interest. Maybe it's helping a little bit. Perhaps there is a significance, but not clinically relevant for that particular patient, so the dropout goes out. What other people say is that part of the placebo effect that we see is done by the interaction with the people. If you have, imagine, I don't know if you ever visited these patients at home. Of course, some of us have, but some of us haven't. But for those that didn't, these patients sometimes are in bed. They have no contact with anybody. They are all day alone. Maybe they have a nurse come in and 1 hour or whatever, they count the pills, help them for a little while, have someone assisting them. So when you go there and see the patient, you are an intervention. Definitely an intervention. And so if you want to be very anal and make sure that everything is okay, and then you call the patient every day, you call them 4 times a day, and the patient is expecting your call, then obviously these patients are going to feel better. So the placebo effect goes up. So what is the answer to that? Then just don't call the patients and just forget about them. And then you have the visits that are more spread, but then the result of that is the compliance may go down because you are not reinforcing the idea that they have to take the pills. Then the dropout rate also goes up. The other way to control that would be with the number of arms that you can make in the study as well. That's the other idea. So the more arms you have, the more chances you have to pick up an effect, because let's say one arm would be placebo, the other arm carnitine 3 grams, the other arm carnitine 2 grams, the other one 1 gram, the other one 500 milligrams. So might be able to pick up a response there. However, the more arms you have into the study, more chances are that you are going to be on the treatment side, right? Maybe you don't get the ideal dose, but you are going to have some. So that generates expectations on the patients. Maybe I'm one of the 3 arms that have the medication, or the 4 arms. Maybe I'm not on the placebo. So that has an impact on it. So in other words, it's just not easy. So it's something that we have to live with. In this study, unfortunately, it really killed us. The bottom line is that for patients that that we accrued and they were fatigued, and we gave them carnitine 2 grams a day, as you can see, the effect is faded away. So what we are doing now is analyzing the subgroup of patients that what they were is carnitine deficient, and see if those patients benefited. And why are we doing this? Well, this is based on the study that we saw before, that there was an indication that it might help. The problem with that is now, even if we see an effect, this is not the main outcome. So it cannot be a positive study. It's still a negative study with a subset of patients that might benefit from it. All right, I think that I bore you guys enough. Torture is over. Thank you.